Your peptide came back at 99.5% purity, the mass spec confirmed molecular weight, every release test passed, and there is still a molecule in that vial. A reviewer can ask you to justify because it weighs exactly what your drug weighs and it comes off the column at the same time. Hello, I'm Raghur Singh Parmar, Director of Scientific Affairs at Resolve Mass Laboratories, and we work with pharma and biotech teams to build peptide impurity profiles that survive regulatory review. Today we are talking about chiral purity, the D-amino acid test most synthetic peptide programs skip until a reviewer asks for it. Before we start, I would like to request you one thing. If you have found anything valuable in this channel, please like and subscribe the channel. This gesture of appreciation really means a lot to us. Let's start. Solid phase peptide synthesis has a quite side effect, documented since not long after Merrifield gave us the technique. Every deprotection cycle exposes your growing chain to base. Base removes the FMOC group you wanted gone and occasionally pulls a proton off an alpha carbon you wanted left alone. When that proton comes back, it can land on the wrong face. You've just made a D-amino acid inside an all-L sequence. The chain keeps growing, the synthesis reports a clean yield, and your new impurity carries the exact molecular formula of your drug substance. So, what does chiral purity actually measure? Total purity by HPLC tells you what fraction of material sits under your peak. Chiral purity asks something narrower. Of every chiral residue in the sequence, how much is still in the L configuration? The answer is never a perfect 100%, and it doesn't need to be. What matters is that you know the number, control it, and hold it to batch to batch. The hotspots are predictable: aspartic acid, cysteine, histidine, and to a lesser degree, serine. The standard purity method can miss a D-amino acid completely. And here is the reason. A peptide with one residue flipped from L to D is a diastereomer of your target. Identical molecular formula, identical exact mass, near identical fragmentation by tandem MS. So mass spectrometry on its own cannot tell you it's there. And because the change is stereochemical rather than compositional, retention on a conventional C18 gradient often shifts by seconds, sometimes not at all. The peak you integrated at 99.5% can be carrying a diastereomer underneath it. Purity and chiral purity are two different measurements. Now, why a D isomer matters? One flipped stereomer matters for three concrete reasons. First, potency receptor binding is a safe recognition, and D residue in a binding region can cost you real activity while your content assay still treats full strength. Second, immunogenicity. A stereochemically altered peptide is a structure the immune system has never seen, and in chronic dose products, that is the risk regulators weigh hardest. Third, comparability. If you're filing a synthetic peptide against a recombinant reference product, you have to show your impurity profile is no worse than theirs. An unmeasured diastereomer is a hole in that argument. Map the risk. Not every sequence carries the same exposure, and you can map this before running a single sample. Aspartic acid followed by glycine and asparagine is the classic hotspot. Base attacks the backbone, forms a 5-membered aspartimide ring, and the ring reopens into beta-aspartyl and D-aspartyl species in one step. Cysteine racemizes during activation. Histidine's imidazole ring does the same to its alpha carbon atom. Glycine has no chiral center at all, so it can't racemize. If mapping your sequence this way is useful, a quick like helps other peptide teams find this. 3 chemistries, 1 signature. Those hotspots produce different chemistry and same analytical signature. The aspartimide route yields up to 4 related species, alpha and beta linkage, L and D configuration, all sharing one mass. Cysteine racemization in a disulfide peptide can scramble the fold without touching the formula. Histidine epimerizes usually appears as a solder that method development quietly optimizes of it. At Resolve, we have worked with sponsors who arrived with a validated purity method and one unexplained solder and left with a chiral amino acid analysis and an impurity story a reviewer could follow. Now, how the test runs. Chiral purity testing runs in 5 stages, and the order really matters here. Stage 1 is a sequence risk map, which residues can racemize and which cannot. Stage 2 is total acid hydrolysis, and this is where labs get burned. Hydrolysis itself racemizes a % or 2, so an all L reference must run alongside every sample so you can correct for it. Stage 3 is chiral derivatization. Murphy's reagent or a chiral thiol with the OPA turning each D and L pair into separable diastereomers. Stage 4 is LCMS or chiral GC to quantify the D fraction residue by residue. Stage 5 is the one people forget, set a specification and support it with the orthogonal intact peptide method. So finally, what should you walk away with this discussion? Purity and chiral purity are not the same measurement. First of all, a D amino acid is isobaric with your API, so mass spec alone will not find it. And a number of you have measured and specifically is always easier to defend than one you haven't. If you are working on an IND, NDA or ANDA for any peptide program, and if you need a CR or CDMO partner for this kind of projects, you can reach out to us, and Resolve Mass Laboratories teams would be happy to support you in these projects. Thank you so much for watching this video till the end. Goodbye for now. Before you go, I want you to do a few things. Number 1, subscribe this channel right now. The more you subscribe, the more valuable pharma insights and expert tips and industry updates we can bring to you. Also, please comment your feedback. This will really help us to improve the content, quality of the videos, and many things. And finally, share this video to at least one person who you think this would really help them. Thank you so much, and I will see you in the next time. Until then, goodbye for now.